I'm Ted Dobie, editor for the journal Neuron, here with Xin Sun, a professor at the University of California, San Diego. Thanks for being with me today.
Well, thank you very much, Ted.
So, as a developmental biologist, how did you begin to become interested in the connections between the lung in particular and the brain?
Yeah, I've been really lucky in terms of getting into this field, really just to follow the findings that we have. So this is about 7 or 8 years ago, we were studying the lung from the developmental biology angle, and one of my students, Kelsey Branchfield, was studying a rare disease called congenital diaphragmatic hernia, and she was particularly looking at a set of genes called roundabout or robo genes, plays a role in that rare disease, and her work found that these genes are expressed actually in neuroendocrine cells, and when they're mutated in those neuroendocrine cells, it leads to activation of those cells. So that's how we got into neuroendocrine cells and thinking about the lung from the point of lung sensing signals coming into the lung. And since the neuroendocrine cells are also innervated, so that's how we pushed off to look at the innervation of the lung, starting from the sensory side, linking onto the signal where it takes off to the brainstem, and now we're looking at the innervation back to the lung cell types. Neuroendocrine cells and many other cell types in the lung receive outputs from the nerve. So that's how we got into it, and it's just been a wonderful journey and learning from immunologist and neuroscientist. So it's a fun journey, and I still feel like an outsider, but I think it's great for others that study the organs. I think there's a lot of places that we can contribute to the field.
So you mentioned neuroendocrine cells, and you mentioned the output back to the lung, and so that relates to the recent findings that you discussed here at the meeting today about particularly about asthma. So can you tell us a little bit about the how the lung senses allergens, and then that affects the phenotype that we associate with asthma?
Yeah, absolutely. So, so this work really builds on many in the field that have carried out to show that sensory neurons, especially TRPV positive sensory neurons in the vagal ganglia, are really important to relay the allergen signal coming into the lung and plays a role in lung response to this. So we pushed off from this work and going further up from the sensory nerves. And so my postdoc, Xin Sun, had done the very meticulous study that she did to find that the NTS, one of the subset of the NTS neurons that expresses DBH, is a key subset that gets activated if we put the allergen to the mouse lung. And so what she continues to find is that if she blocks the activation of those neurons, she's able to block lung response to the allergen. Not all the lung response to allergen, but a specific type of airway constriction response, which also speaks to the specificity of what's the neural circuit involvement in the allergen response. And what she was able to then find is from those NTS DBH neurons, there is a relay through another node called the nucleus ambiguus in the brainstem, which then comes to postganglionic neurons in the lung, which then goes into the particular airway and regulates the smooth muscle constriction of the airway. So altogether, I think from our work and other works, we've now identified a full neural circuit that starts from the lung to the brainstem and back, and there are at least 6 nodes in that neural circuit that plays a role in this process. And I think the other exciting part is that we're also starting to identify the signals from one node to another. So for example, the node from the NTS neurons to the NA is mediated through norepinephrine. So this then potentially open up neuromodulation for asthma response.
Right.
Yeah.
And do you see these results helping us also understand and apply new therapies towards upper respiratory infections, exercise-induced asthma, these sorts of things?
Yeah, absolutely. So in the future, these are definitely future directions that we want to get at. My lab studies mechanisms of lung diseases. So in terms of infection, it's one of the major things that we study. There are some really beautiful work from Steve Liebowitz' lab and others, which are already getting to flu induction and how that interfaces with the nervous system. So that's an area that I think we as a field are going towards and will respond. So in terms of exercise-induced asthma, we have no idea whether that the neural circuit plays a role, and I would not surprised at all. So that's one of the directions I think as a field we should go to. But we're also really interested in terms of the big picture of the lung as a sensory organ, looking really at baseline in the healthy lung. Because it has huge surface area, the lung does, and has a lot of inputs of aerosolized signals. What might be the specificity that is induced, and it's all unconscious reception of those signals, and from those specific detection of the signal, what might be the output that the nervous system play a role in. So there'll be a huge area of investigation.
So this I would say interoception generally in the brain-body connection is a fairly young field, and so now having worked in it for a few years, are you learning any lessons from the lung, from other organs that you see kind of generalizable rules emerging?
That's an excellent question. I think the very astounding message from this conference is that the nervous system sense interoception is very pervasive. It's just everywhere. So I think that's a very clear message. In terms of the rules, I think we're only starting to understanding it. So there are a few speakers here talked about food allergens, and we study the aerosolized allergen. There are some immune crosstalks, but I think there are also some differences in terms of the different sensory pathway as well as the output pathway. So it will be really interesting to study both on the positive regulation side as well as some of the feedback negative regulations, I think.
So kind of any final thoughts on where you see lung physiologists, respiratory therapists, these sorts of folks kind of implementing what we're learning about how the brain and lungs connect to kind of advance the field overall?
Yeah, so a really good question. So I think I'm actually in the pediatrics department, and many of my colleagues at UCSD are clinicians. So when I tell them about our study, that the nervous system actually is very intimately involved in this, they're very excited. In terms of asthma in particular, the current therapies are inhaled steroids, so lots of side effects. So they are excited in implementing some of the more specific neuromodulatory ways to control those diseases. And I think we're getting close to that step.
Do you think vagal nerve stimulation is a viable thing for asthma?
So I think the next step is to make it more specific, right? So figuring out the specific type of nerves that we can stimulate and what's the duration and amplitude.
So you said you work with a lot of pediatricians, and I know you work with a lot of, in terms of rare disorders. How have rare disorders been particularly helpful in kind of understanding the lung and how the lung functions? So
great question. So I am have a very close connection to rare disorders because I'm also trained a geneticist. So the rare disorders really highlight, a few triggers, single gene or other triggers. So it really lead us into the mechanisms that are clean, that we can dissect very clearly. And then oftentimes these genes are also altered in more common diseases, right? So they might, in those settings contribute 5%, 10%. But with the knowledge from the rare disease, we can then ask how does this pathway as a 10% contribute to complex diseases.
Great.
So,
well, thank you so much for joining me today, and I'm excited to see the developments in this field, probably that you'll be leading the way on.
Thank you very much.
Okay, thank you.